Peace upon all my dear students. Today we are going to start our first lecture in the course of histology, or histology and cell biology, or histology and cytopathology. All these courses should be begin by this essential lecture. As we usually have to start, we will start by learning outcomes. Learning outcomes. By the end of this lecture, you will be able to define histology, to compare between different microtechniques, list the steps of paraffin technique, and differentiate various staining methods. I'm dealing with the following contents: introduction to microtechnique, steps of paraffin technique, and tissue staining by hematoxylin and leucine, and finally we are going to special stains. Introduction to microtechnique. Introduction to microtechnique, we are going to why microtechnique? Microtechnique, it is a technique used to study tissue and cells. What is the cell? Cell is the basic structural and functional unit of all living organisms. Cytology or cell biology, it is the science that deal with the study of the cell, or study of the biology of the cell, how the cell live, how the cell grow, how the cell perform its function. While histology, it is the study of the fine details of tissues using microscopes. Specimens of tissues are prepared using special processes. We call it histological techniques. So, how we could study the cell, how we could study the tissues? We study the cell and we study the tissues through different techniques. These techniques, we call it histological technique. Microtechnique, so they are different methods used to prepare histological sections for microscopic examination. These include preparing section, staining of the section. Here you have different methods. Different methods here include many methods, many techniques. Preferably, I divide the microtechnique into two parts. Preparation of section. From the target tissue, I will prepare a section, and then I will stain the section. And if we prepare a section from the tissue and put this section or this slide under the microscope, you will find nothing. You didn't find the tissue. The tissue or the section needs to be stained, to be fully punctually examined. Yes. Methods of preparing sections for microscopic examination. What are the methods for preparing sections for microscopic examination? Paraffin technique is the most common. Freezing technique is the most rapid or very rapid. And plastic embedding sections, which is electron microscopic sections. It is the electron microscopic sections. Let's start by steps of paraffin technique. Steps of paraffin technique. We have to start correctly the steps of paraffin technique or the steps of any technique in histology. The best start here is the measurements used in histological study. Histological study, when you deal with the microscopic examination of tissue, microscopic examination of the cell, you have to know the units of measuring cell and tissue. The units, we measure the cell or we measure the tissue usually by this, by micrometer. Micrometer is 10 to the power of minus 6 of the meter. 10 to the power of minus 6 of the meter. If we get larger, a little bit larger, 1 millimeter, it is 10 to the power of minus 3 of the meter. And if we get more larger, 1 centimeter, it equal 10 to the power of minus 2 of the meter. 1 over hundredths of the meter, 1 thousandths of the meter, 1 millionth of the meter, which is the micrometer, the most commonly used unit here. If you ask me, could we get more smaller? Yes, if we get more smaller, you will get the unit 1 nanometer. 1 nanometer equal 10 to the power of minus 9 of the meter, and so on and so on. If you ask what is the relation between the millimeter and the micrometer, you will find that the millimeter equal 1000 micrometer, and 1 micrometer equal 1000 nanometer, and you will get, and you can get many relations between different units. Don't forget that the most common unit used here is the micrometer, which equal 10 to the power of minus 6 of the meter. Paraffin technique. Paraffin technique, it is the most common technique, and we call it the routine method. The idea of the paraffin technique is to be able to cut the tissue into thin sections and preserve it. This is the idea. This is the idea of paraffin technique. If I give you a tissue and ask you to examine this under the microscope, you will say to me, I can't examine this tissue under the microscope except I cut it into very thin sections, preserve it, and finally I will stain it. By these 3 conditions only, you can examine the tissue under the microscope, and you can start your histology study. How we could accomplish this? To accomplish this, the tissue must be hard. The tissue must be hardened to be cut. If the tissue soft, you will not be able to cut this tissue. And how to get this tissue hard? You have to replace the water in the tissue by the hard substance. We call it paraffin substance or paraffin wax, and so we call the technique paraffin technique. Steps of paraffin technique. The steps of paraffin technique, 8 steps. 8 steps. Each step had certain procedures, certain rules. Obtaining of tissues is the first step. Fixation of the tissue, dehydration to remove water, cleaning to remove the alcohol, impregnation is to replace the xylene by paraffin wax, embedding is the formation of hard block. Then I will start cutting of the hard block, and then I stain the tissue. This is the 8 steps. This is the 8 steps of paraffin technique. Could you notice these 8 steps to be familiar with them, starting from obtaining tissue until sections, putting sections into the glass slide ready to be stained, Obtaining of tissue, or we call it tissue sample. Obtaining of tissue, the tissue should be small piece, about 1 centimeter, no more than 1 centimeter, and it should be taken immediately, and must be fresh, immediately after death, immediately during surgery, or biopsy taken, should be obtained rapidly and processed rapidly. The aiming of fresh and immediately taking the slide to prevent the action of intrinsic enzyme and extrinsic bacteria, as we will discuss in the next step. Next step is the fixation. Fixation, here I have to fix the tissue. Fixation of the tissue is to prevent autolysis through destruction of enzymes that cause post-mortem degeneration, and this is one of the enemies that disturbs the tissue. Number two, prevent putrefaction by killing the bacteria, and this is the second enemy. These two enemies are the enemy of the living organism. So when we take the biopsy outside the human body, or when we take the tissue outside, it will be attacked by intrinsic enzymes and extrinsic bacteria. Aiming of fixation is to stop these two enemies, destroy the enzymes, killing the bacteria, and hardening the tissue, and this will make it easy to be cut, and mordanting effect, and this increase the affinity of the tissue to stay. Fixatives. Fixatives, they are chemical substances used to preserve the tissue. They are chemical substances used to preserve the tissue. There are many tissue fixatives. Buffered formalin, buffered glutaraldehyde are the most common fixative. But there are many types of fixatives beyond this scope of the lecture, beyond the scope of this lecture. So we have to memorize and recognize these two most important fixatives. Number three, dehydration, to replace the water in tissues by alcohol. You know that the most common chemical component of any living organisms is the water. The process of microtechnique and the process of fixation includes the removal of the water from the tissue and replace the water by the alcohol. It is done by passing the fixed tissue in ascending grades of alcohol, 50%, 70%, and then we put it into 90% until we reach to absolute alcohol. You will note that the degree and the concentration raises from 50% until absolute. If you think about the cause, you will find that the concentration of water here is 50% and the 50% alcohol. Then we minimize the concentration of water into 30%. Then here the concentration of water becomes 10%. And then finally the water content will be zero concentration. By this concentration, the only chemical component present inside the tissue is the alcohol. Dehydration should be gradual. Why? Yes, to minimize shrinkage of the tissue. If you imagine you put the tissue within the absolute directly, you will get shrinked tissue and distort the shape of the tissue. Step number 4 in microtechnique is during dehydration of water in the tissue, we have to replace the alcohol by paraffin. But when you get to replace paraffin, to put the paraffin to replace the alcohol, you will find that the paraffin wax not alcohol soluble. What is the meaning of that? The meaning is that the paraffin cannot replace the alcohol. Both of them not compatible with each other. So the scientists of histology try and search for a certain chemical structure that can be miscible with the alcohol and miscible with the paraffin could deal with the alcohol and the paraffin could deal with it. This magic substance is the xylol or xylene, and this step we call it clearing as the xylol or xylene replace the alcohol and the paraffin will replace the xylene. But moreover, you will find that the xylene is essential also in keeping and making the tissue transparent, and this transparency will allow the light examination or the light microscopic examination very easy. So to summarize the function of the xylene is miscible with the alcohol and miscible with the paraffin, and the second step, our second function of xylene is makes the tissue transparent, Fifth step here is the impregnation in soft paraffin. This is allowed to occur at melting point of temperature. Impregnation in soft paraffin. When I said soft paraffin, to infiltrate the tissue by paraffin, this should be occurred in an oven with degree of temperature 55 to 60 degree centigrade. This degree of temperature, the paraffin will be in a liquid form, so I call it liquid paraffin, and the liquid paraffin infiltrates the tissue and replace what are the chemical components within the tissue at this moment? Right, it is the xylene or the xylol. Next, let's look at this picture. This picture contains the oven with the degree 55 to 60 degrees centigrade, xylene here in a liquid form, and xylene here replace the paraffin here. Paraffin here will replace the xylene. Paraffin here in a liquid form at the 55 degree. The wax will be in a liquid form and it will infiltrate the tissue. And replace the xylene. The sixth step in microtechnique is the embedding in hard paraffin. Please revise with me the steps. Number 1, obtaining of tissue. Number 2, fixation by formalin. Number 3, dehydration to replace water by alcohol. Number 4, clearing. And number 5, impregnation by soft paraffin. Here we came to the sixth step, which is embedding in hard paraffin. Remember that the melting point of hard paraffin is 55 degrees centigrade. Embedding, how I could embed? I embed the tissue by putting the tissue into cassette containing hard paraffin. Paraffin harden as it cools, forming the paraffin block. What I will do with the paraffin block? The paraffin block is hard, and so I can section this block. I can section this tissue into very thin sections, as we see here. This is the cassette, and I put the tissue in the cassette. Then I will pour the soft paraffin, and then I will leave the cassette and the paraffin to cool. After cooling of the paraffin, I can extract the tissue block. As you will see, this is the block, another block. Notice the tissue within the block This is a third block, fourth block, and so on. You will find the tissue in a brown color within the wax in the white color. Number 7 in the microtechnique steps is the sectioning. Sectioning, here you have to know this instrument. This instrument is called microtome. Microtome. It is the tool of sectioning the tissue. We use this microtome to cut the tissue into thin serial sections. Thin serial sections. What I'm going to cut here, I will going to cut the block which I prepare from the previous steps. The block contain formed of wax and contain paraffin. Paraffin and tissue, hard tissue. I'm going to cut the tissue into very thin sections. What are the thickness of the sections I'm going to cut? Yes, it is from 5 to 10 micrometer. From 5 to 10 micrometer. Don't forget this figure, please. It is very important. It is the most common thickness used in studying histology. Let's see the technique. The technique of sectioning. The technique of sectioning, you could find it in many YouTube. Many YouTubes contain the technique of sectioning. Here, the doctor or the technician here used the microtome to section the block into thin serial sections. As you see, thin serial sections. This is the wheel of the microtome, and this is the thickness we require, usually from 5 to 10 micrometer. You remember the micrometer equal 10 to the power of -6 of the meter. Then I will put the block and start to cut the section into thin serial section. I hope it will be clear. After that, I will put the serial sections into water path adjusted at 37 degrees centigrade or warm water. This will allow the thin sections to float and to remove the folding of the sections. Mounting is done by putting the section on a slide, which becomes ready for staining. You see here, I mount the section by the slide. I mount the section by the slide, and this slide now I will put it into a warm hood to evaporize the water, and this moment the section or the thin sections on the slide ready for what? This is my question. Ready for what? This section is ready for what? Ready for to be examined by the microscope, or it should be stained? Yes, it should be stained. So the section here is ready to be stained by stains of histology, as we will discuss later. At this point, I have to stop and think about the advantage and disadvantage of paraffin technique. What are the advantage and disadvantage of paraffin technique? Advantage of paraffin technique takes short time. Paraffin techniques give very thin serial. What is the meaning of serial? What is the importance of serial here? Serial means I have many steps, many sections. So I have plenty of sections to be stained, to be examined. So I will get a reserve from the tissue to be examined, to be studied. And the thin paraffin sections can be stained easily. This is easy technique, short technique, create many sections. Disadvantage of paraffin technique, it is a minimal disadvantage actually. Paraffin technique cannot be used to stain the fat. The fat will be removed by the chemicals we use. We use here xylene, we use alcohol, we use the alcohol and the xylene remove the fat from the tissues. So I will find no fat within the tissue. Also, raising the temperature in the paraffin technique, to which degree? Do you remember? Yes, excellent. We raise the temperature into 55 degrees centigrade to 60 degrees centigrade. So this will remove or denaturize the enzymes. So I couldn't examine the enzymes within the tissue due to the heat of the microtechnique, paraffin technique. The paraffin, we raise the temperature into 55 degrees, so the enzymes will be masked, will be denaturized or lost its sequence and lost its polypeptide sequence and amino acid sequence, so I can't examine the enzymes. So these two disadvantage of paraffin techniques. Next video I will explain the tissue staining by hematoxylin and eosin. This is the next step, and the special stain. Thank you for watching the video. I hope you get the benefit of this video. You can ask me any questions and I'm ready to answer all your questions. Have a nice day. Many thanks for all. Goodbye.